Welcome all. I'm Dave Margolis from the University of North Carolina with Rémi Fromentin from University of Montreal. We're happy to chair this session, Undetected HIV under HAART. I assume we'll get some more influx of people as the time goes on, but I thought we should get started. And just to sort of frame the discussion, we have a number of interesting talks today. I thought I'd show you this version of the conceptualization of assays of virus. This data really displaying virus, evidence of virus recovered from circulating cells. This has been discussed several times at the meeting, but I think it's quite relevant to some of the talks we'll hear about. It's quite simple to quantitate HIV DNA in circulating cells, and it's much more difficult to quantitate replication-competent virus emerging from circulating resting cells. And there's a large-scale difference between the two measures. And as we heard at several points in the meeting, this triangle of replication-competent virus is probably this underrepresentation. If one does repeated stimulation, you could probably double or triple the size of that triangle. More recently, attempts to make easier, better, more tractable assays have, in the literature, resulted in the reporting of a number of different assays measuring HIV RNA, either associated to cells in circulation, in cells that have been stimulated outside of the body, or being produced by cells put into culture. And in fact, I believe that just maybe a week and a half ago, Vicente Planel's group published yet another version of this assay, of an assay. And as you can see, that is another representation of persistence, but perhaps an overestimation of it. So the real reservoir of persistent replication-competent virus is somewhere in between. We have difficulty measuring this. I would propose, and probably a lot of other people agree, that if we had a tractable, sensitive measure of HIV antigen, cells that produce antigen, that might be another useful tool, particularly as we might not mind if we just cleared all the cells that made HIV antigen, because that might have other benefits for our patients. So this is really the state of knowledge of cells taken from the blood or samples taken from the blood. But certainly, as we know, tissue is a very important site of HIV persistence, and all of these questions become much more complicated when you leave the blood and try to enter the tissue. So we have several speakers today dealing with different parts of this issue, and I'll start by asking Chaired Professor Catherine Lizarriaga from the University of Massachusetts, Professor of Molecular Microbiology, Pediatrics, and Medicine, and an old friend. Thanks very much.